Poplars are fast-growing, early successional species that tend to grow in rocky or sandy substrate. Common poplars include black cottonwood, Populus trichocarpa, and Sitka willow, Salix sitchensis, and they grow in riparian zones in western Washington. Poplars are known to fix nitrogen via nitrogen-fixing microbes in their stem tissue. The endophytes found in poplar stem tissues do not cause disease or any other harm to poplar plants, but rather are very beneficial. They provide hormones, peptide antibiotics, and other substances that benefit the hosts. They are thus classified as plant growth promoting bacteria. These bacteria are known as diazotrophic bacteria, and they are large endophytic communities. Although their function is still being researched, it is linked to diatomic nitrogen fixation, thus can explain poplar's fast growth in nutrient-poor environments. Research has verified their nitrogen fixation by using polymerase chain reactions to code for the presence of a gene that encodes for nitrogenase, which is a nitrogen-fixing facilitating enzyme. In the middle of nitrogenase is a unique complex of iron, sulfur, and a molybdenum ion. For our purposes, I'm going to refer to the structure as its chemical element name, MO, which is thought to perform the nitrogen-fixing reaction. A series of cofactors feed electrons to the MO-Fe cluster. Electrons start at a pair of ATP molecules. Then they flow inwards into the iron-sulfur cluster, then to the P cluster, and finally to the MO-Fe cluster. The MO-Fe cluster is at the bottom, the MO atom in bright red, and then a molecule shown with white carbon atoms and pink oxygen atoms help to stabilize this metal ion. The P cluster is in the middle, and the iron-sulfur cluster of the Fe protein is at the top. In spite of the detailed knowledge provided by the structures of nitrogenase, the actual binding site for nitrogen gas is still subject to controversy and intense study. There have also been studies conducted to look at the less common N15 isotope present in these bacteria, and the bacteria were shown to fix 20 times more of this isotope than the more common N14 isotope. Also, when poplars were experimentally inoculated with this bacteria, they increased in weight by 42% and in nitrogen concentrations by 37%. This has huge implications for renewable energy crops because poplars that have a symbiotic relationship with this endophyte can produce large amounts of biomass without much need for fertilizer, which is important in agroforestry. It is also an opportunity to use poplars to sequester excess nitrogen from sites that have a surplus of nitrogen.